Hi, my name is Timofey Prodanov, my co-author Vikas Bansal. We are from the University of California, San Diego, and we would like to present you a robust and accurate estimation of copy number for duplicated genes using whole genome sequencing. Human genome contains a large number of segmental duplications. Segmental duplications come in different sizes. For example, duplications longer than 3 kilobases with sequence similarity over 97% cover over 100 megabases of the human genome and around 1,000 protein coding genes. And 120 of these genes are disease associated. Segmental duplications, especially long and with high sequence similarity, are problematic for short read sequencing. One of the problems is that read mappers don't know where to put reads, so this leads to downstream problems, for example, copy number variation detection or single nucleotide variation detection. Its size is skipped in duplicated regions, or it can be incorrect. One of the duplicated genes is SMN1. Its second copy is SMN2. They lie within a 200 kilobase duplication with very high sequence similarity. And SMN1 is one of the most studied duplicated genes because mutations and copy number changes in it can lead to spinal muscular atrophy. And what is interesting is that paralog-specific copy number matters, meaning that if an individual has 3 copies and 2 of them are SMN1 and 1 of them is SMN2, this would be a healthy individual. And the other way around, if there is 1 copy of SMN1 and 2 copies of SMN2, this can be an affected individual. There is a useful definition in segmental duplications. It's paralogous sequence variants, or PSVs. And PSV is a small difference between repeat copies. This can be one or several nucleotides. And PSVs are the main thing that we use to distinguish between copies. So, for example, here in a specific position, copy A has T, but copy B has G. And the problem with PSV is that they can be unreliable. So reliable PSVs would have 2T and 2Gs, but it's possible that we observe 4Ts in this position. Finally, we want to define what copy number is. Suppose there is a n copy duplication. Then we can define paralog-specific copy number as a tuple of length n, which is copy number of each individual copy. For example, sample 1 here has PSCN 2,2, meaning that it has 2 copies of each repeat copy. Aggregate copy number is a sum over PSCN. It's possible that a copy is missing. For example, PSCN here is 2,1 for sample 2, and aggregate copy number is 3. The other way around, copy can be extra copy. For example, sample 3 here has 3 copies of copy A. You can notice that its aggregate copy number is the same as sample 1. We would like to present you a tool, Parascopy. It takes whole genome sequencing for multiple samples as input and also a list of regions of interest. For each region, Parascopy will find aggregate copy number and parallel copy number profile. And here you can see a pipeline of the tool. First, we start by finding all the repeat copies of the region of interest and all the PSV positions. And we do that by looking at a pre-computed table of duplications. Next, for each sample, we estimate background tree depths. We do that by looking at non-duplicated regions. We do it independently for different samples. And also, we estimate different background tree depths for different GC content values. And I will explain next steps in more details. We start by remapping reads from all the copies of the duplication to the first copy. And we do that because we don't want to depend on the read mapping and read mapping quality. And as a result, we will obtain aggregate read depths and read depths for different alleles of the PSVs. We use aggregate read depths as input to the hidden Markov model, and the hidden Markov model will provide us with aggregate copy number estimates. And because we have multiple samples, we can use it to refine HMM parameters. For example, refine initial probabilities and transition probabilities. Let's say we have many samples that have a deletion close to each other. We can use that information to increase transition probability in that position, and it may help us to find more samples with a deletion. So in this example, 3 of the samples have constant copy number, and it's easy to identify them, but last 3 samples have a deletion at the end. And on the first HMM iteration, on the right, you can see that only 2 of the samples were identified as having a deletion. And on the last iteration, after we refined parameters and refined transition probabilities, we identified all 3 samples as having a deletion and all of them at the same position. After we have aggregate copy number, we want to identify paralog-specific copy number. For example, here, let's say we have 3 samples with aggregate copy number 4, and we have PSV allelic read depth. So yellow color shows number of reads that support allele from the first copy. Blue shows number of reads from the second copy. And the problem is that if you look at different PSVs, they will say different things. For example, PSV2 will say that all the samples have many copies of copy 2. But this is because PSV2 is probably unreliable. Additionally, if you look at sample C, and we would want to identify unreliable PSVs, we would say, okay, all the PSVs are unreliable because ratio between allelic read depths is not 1:1. To solve this problem, we run an EM algorithm. We want to identify paralog-specific copy number for all samples, and we also want to find reliable PSVs. We defined it as a F values, frequency of the reference allele for each copy. And what is important to understand is that if a PSV is reliable, all its F values will be close to 1. So in this example, PSV2 has F1 not close to 1 because it's unreliable. Additionally, we ran all the analyses for multiple samples. If we want to analyze new samples, we allow to save model parameters and then load them back to analyze new samples. It will be much faster than to rerun everything for an extended set of samples. We analyze different loci independently. Because of that, we can work in parallel. So for 700 samples at 167 loci, it took 19 hours using 16 threads to find copy number. And if you analyze in 2 samples using pre-computed model parameters, it will take at maximum 25 minutes. On this plot, you can see Parascopy estimates of the aggregate copy number profiles for around 500 samples from the 1000 Genomes project. This plot shows SMN1-2 duplication, and it is already known that SMN2 has a common deletion at the end at exon 7 and 8. And without prior knowledge about this deletion, Parascopy detects it, and the position of the deletion roughly matches position that Parascopy detects. Next, we wanted to validate Parascopy results. Here you can see we looked at MLPA experimental data. It provides 2 observations for each sample, aggregate copy number for exons 1, 6, and aggregate copy number for exons 7 and 8 because of the common deletion. And across 970 samples that we looked at, we have 100% concordance. We looked at more disease-associated genes. Here you can see 4 of them. And we looked at 3 methods. So on the right, you can see Parascopy. Next, there is QuickMare2, which provides paralog-specific copy number across the whole genome. And then there is custom methods that are designed specifically for one gene. For example, for SMN1, there is one method. For SRGAP2, there is another method. And if you look at this, in SMN1-2, Parascopy has perfect results, but so does another method. QuickMare2 have has lower accuracy compared to Parascopy or a custom method. For SRGAP2, Parascopy and custom method have almost perfect accuracy for aggregate copy number, but for paralog-specific copy number, Parascopy has better results than a custom method. QuickMare2 has lower accuracy than Parascopy in both cases. Next, for C4A, we looked only at QuickMare2 and Parascopy has higher accuracy than QuickMare2 in both aggregate and paralog-specific copy numbers, but you can see that paralog-specific accuracy is not 100%. Next, we looked at AME1C. For this gene, aggregate copy number varies a lot across the population. It can be 2, it can be 18. And so we looked at correlation coefficient. And you can see here that Parascopy provides better correlation coefficient than other methods. Next, we looked at PSVs in SMN1 and around it. On the y-axis, it's frequency of the reference allele. It's F values that we defined earlier. If PSV is reliable, points will be high on both subplots. Overall, there are 10 to 19 reliable PSVs that we can detect with Parascopy across 3 populations. However, there are no reliable PSVs in African population, which is consistent with previous studies. There is a custom method specifically designed for SMN1. It uses 8 reliable PSVs, and they are shown in red. And Parascopy detects all of them, but additionally we detect some other PSVs depending on the population. Next, we look at the percentage of reliable PSVs across 59 disease-associated genes. Some of the genes have very low percentage of reliable PSVs. For example, on the left you can see PMS2, SMN1. These are very important disease-associated genes, but they have low percentage of reliable PSVs. And this is important because other methods that don't look at reliable PSVs, they will make errors there. For example, read mapping can be incorrect because there is unreliable PSVs next to a read. And additional point is that if you look at different populations, you can see that percentage of reliable PSVs is often very similar across different populations. Auto-A is another disease-associated gene. It is related to hearing loss, and in the reference it has 2 copies, Auto-A and pseudogene Auto-A-P1. So the aggregate copy number would be 4. However, as you can see from the plot, most common copy number is actually 6, so there is a missing copy in the reference. We looked at a novel CHM13 assembly, and it has 3 repeat copies. And what we can do with it is we can take a third copy and we can add it to our database of duplications. And by doing that, we can then see that we can now detect paralog-specific copy number for all 3 copies, not only 2 that were present in the original reference. Finally, we look at the robustness of Parascopy, which means that we looked at the same samples but with different inputs should provide similar results. So here we looked at 2 independent sequencing datasets for 83 Han Chinese samples, and we want to see that aggregate copy number and paralog-specific copy number is almost the same. We looked at 167 duplicated loci. If you look at red line, it shows all of these loci, and y-axis percentage of loci, and x-axis is concordance threshold. For a reasonable concordant threshold, more than 95% loci are concordant. However, if we limit the loci and only take loci with low copy number, such as 2 repeat copies or 3 repeat copies, percentage of loci that are concordant across two sequencing datasets now close to 100%, if you look at green and cyan lines. Finally, if you look at paralog-specific copy number, shown in purple, it's almost 100%. But here we can say that we don't detect paralog-specific copy always. For example, if there are no reliable PSVs, we skip this region. So whenever we assign paralog-specific copy number, it would be the same across two independent datasets. Finally, we can say that Parascopy uses multiple samples to estimate copy number. Then we find reliable PSVs and use them to estimate paralog-specific copy number. Parascopy has higher or sometimes equal accuracy compared to other sequencing-based methods. And Parascopy can analyze large number of duplicated loci. We are not limited to a single region. This loci can have diverse repeat structures. For example, reference can have 3 repeat copies, then it's 2 repeat copies, and it's again 3 repeat copies. Parascopy can work with that. You can find Parascopy on the link below. Thank you for listening.